Hello everyone, welcome to the new session of Biological Classification NCERT exercise. So here we are going to solve some of the questions from the NCERT, right? We have already done some of the questions and the rest of question we are going to solve in this particular session. So let's start. So plants are autotrophic. We all know that plants are autotrophic. They can make their own food by the process of photosynthesis. Now, can you think some plants that are partially heterotrophic? Means sometimes they can do the process of photosynthesis and they are autotrophic, but sometimes they cannot make their own food. So they are doing the process, taking the food as a heterotrophic mode of nutrition, right? So that are called as partially heterotrophic. So can you think some of the plants that are partially heterotrophic? So here comes the solutions. While plants are primarily autotrophic, meaning they produce their own food through the photosynthesis. Yes, they can make their own food through the process of photosynthesis. There are some plants that exhibit partial heterotrophy, but yes, there are some plants that cannot make their own food. So they exhibit partial heterotrophy phenomena. Yes, and either by obtaining the nutrient from the external source or by supplementing their photosynthetic capacity with the heterotrophic strategies. Okay, so either they obtain nutrient from the external source or supplements their photosynthetic capacity with their heterotrophic strategies. So here are some of the examples. Now the first one is the epiphytic plants, the plants that get attached to the other plants, right? Mostly the trees. Okay, so let us see what are the epiphytic plants. So epiphytic plants such as a certain species of the orchids and bromeliads grow on other plants or the structures but do not parasitize them. They grow on them, okay, but they are not taking food as a parasitic mode of nutrition. They just get attached to it. Okay, they grow on them, and these plants typically derived water and minerals from the air. So they are taking water from the air only. Whatever the atmospheric water vapor is there, they take that from the air and rainwater or decaying organic matter accumulating around them. So they are attached to the other plants, right? Or that could be a branch, that could be a main, you can say, the stem of the tree. Okay, so they are attached to it, and when the bark is decaying, they are taking that organic matter. Okay, and they are accumulating that around themselves. So while they may still perform the photosynthesis, they are actually green in color. They are having the leaves. They can do the process of photosynthesis. They rely partially on the external source of the nutrient, but still they rely partially on the external source for the nutrients. So this is one of the epiphytic plants. Now, mycoheterotrophic plants form a unique symbiotic relationship with the fungi. Hmm. And from which they obtain the organic carbon and nutrients. So these plants lack chlorophyll and cannot do the process of photosynthesis. So they are relying entirely on their fungal partners for these substances. Now, examples include certain species of orchids, such as members of the genus uh, Corallorhiza, and some monotropa species, Indian pipe or the ghost plant. Okay, so this is one of the very important plant, ghost plant. It is also known as ghost plant. So this mycoheterotrophic plant is another example of this. The third one is the carnivorous plant. Carnivorous plants supplement their nutrients uptake by capturing and digesting small animals. They can capture or they can digest the small animals, typically insects and other arthropods. So their body or their body is organized in such a way that they can capture the insects, they can capture the flies, and they can digest themselves in their digesting system. They are having the particular digestive system. They are having a secretion of many enzymes, okay, and chemicals that can degrade or digest these animals. So while they still perform the process of photosynthesis, carnivorous plants have evolved specialized structures such as modified leaves or traps to attract, capture, and digest the prey. Their leaves are generally modified, okay, so that they can trap the insects. And actually, they are doing because they are growing in a nitrogen deficient environment. Means the soil is not having any nitrogen in there, but still they are growing. So how, and you all know that the nitrogen is so very important for the process of like protein synthesis. In the protein synthesis, nitrogen is required in a good amount. So they take that they fulfill that nitrogen requirement by capturing these insects, as these insects are having a good amount of nitrogen in their body, right? So common examples include the Venus flytrap. This is one of the insectivorous plant, and scientific name is Dionia muscipula, and a pitcher plant, very common, Nepenthes species. And the another example is the sundew drosera species, and another one is the bladderwort, that is the utricularia species. So these are some of the carnivorous plants that do follow the heterotrophic mode of nutrition. Another is the mixotrophic plant. Mixotrophic plant. So some plant exhibit a mixotrophic lifestyle, meaning they can derive energy and nutrient from both the autotrophic and heterotrophic sources. They can derive energy, or they can take their nutrients from both autotrophic as well as the heterotrophic sources. So mixotrophic plants may engage in the process of photosynthesis but also supplement their nutrition by capturing prey for the mycorrhizal association with the fungi, or utilizing the organic matter from their environment. So what happened? There are some higher trees. Their roots are going to the many far away, and on their root surface some fungi are growing, making a mat over its root surface. That is a mycorrhizal association, that is showing the mycorrhizal association, right? Now these tree, what they do, they provide some nutrition to the fungal component that is forming the mycorrhizal association, and in return these fungi is giving some very important minerals that that they these plants cannot make available for them. So what they do, these fungi take away minerals from far away places because they are spreading to very far away places, and they are having those minerals and making available to these trees, okay, and in return these are getting some sort of a food material. So examples include certain aquatic plants like bladderworts, utricularia species, which can perform both the photosynthesis and capture prey through the specialized trap. So these are also one of the example, that is the bladderwort utricularia species. Okay, here comes the next question. What do the term phycobiont and mycobiont signify? Right. So these are the two terms that is the comes under the, when you study about lichen, then you come to this particular terms, that is the phycobiont and mycobiont. So let us see. Phycobiont. The term phycobiont refers to the photosynthetic partner in the lichen symbiosis. So lichen is an association. It is a type of association, and that type of symbiotic association. Symbiotic association means both the component, algal component as well as the fungal component, they come together and live in an association, and that is the symbiotic association that is called as lichen, right? Now here the phycobiont is doing the process of photosynthesis. So it is a component responsible for producing the organic carbon through photosynthesis, which provide energy and nutrient to the lichen symbiosis. So this one is the algal is having the chlorophyll, right? The algal partner is having the chlorophyll. They are having the chloroplast, and inside that they are having the chlorophyll. Now this chlorophyll, due to the presence of this chlorophyll, they are able to capture the sunlight, and they can make their own food. and that food is made available to the fungal component as well, right? So the phycobionts are typically green, that is the chlorophyta or cyanobacteria. So some cyanobacteria may also be there, that is a monera, right? And in if you talk about the algal component, then in the algal, generally the green algae, that is the chlorophyce or the chlorophyta is there. And although sometimes they may include other photosynthetic organism as well as the lichen thallus body, the phycobiont cell are embedded within the fungal matrix. So this algal component is totally inside the this captured all over or covered by all over by the fungal matrix. Okay, it is placed very inside to the lichen thallus, and where they are protected and provided with a suitable micro environment for the growth and photosynthesis, because they are like they cannot tolerate such a high intensity of the light, right? So these fungal component make them available or make keep them very inside the surface of the fungal component, right? They are having a upper covering with the made up of the fungal component, and below that there is an algal zone where these algae are kept. So that is making a suitable micro environment for them, and that is very very good environment for the algal component, and there they can do the process of photosynthesis. Okay, so the phycobiont benefit from the association by receiving the water and minerals and protection from the environmental stress. This is very very important, because the protection is provided by the fungal component, right? Because they are making this keeping this algal component deep inside, and not letting them allow to expose to the environmental stress. So, while providing the microbiome with the organic carbon compound synthesizing through the photosynthesis, so these algal component, they get the organic carbon, that is the organic carbon compounds, and they can synthesize glucose or the food material through the process of photosynthesis. Now comes the microbiome, the fungal component. The term microbiome refers to the fungal partner in the lichen symbiosis. So, if we talk about the fungal partner, that is the fungal partner, that is the microbiome in the case of lichen symbiosis. It provides the structural framework and protection for the lichen. So, they are providing the protection to the lichen, and as well as facilitating the exchange of nutrient from the phycobiont. So they are helping this phycobiont to get the exchange of nutrients. Okay, and they are also protecting, because they are keeping this phycobiont deep below the surface of the fungal component. So microbiome belongs to the various fungal taxa, including the Ascomycota, Basidiomycota, with the Ascomycetes being the most common group associated with the lichen. Okay, so we know that the Ascomycota and Basidiomycota are the two fungal taxa. So according to the lichen which we know today, the Ascomycota is the most common type of fungal component today we can see in the case of lichens. The microbiome forms the bulk of the lichen thallus and providing a suitable habitat for the phycobiont and contributing to the lichen's overall morphology and ecological adaptation. So, this is the morphological and ecological adaptation that is provided by these lichens. Okay, so this microbiont is very very important, and this is making the bulk of the lichen thallus. So main bulk of the thallus is made by the fungal component, that is the microbiome, because there is an only one single zone where this phycobiont is there. Okay, the rest is the microbiont. In return, the hosting the phycobiont and providing the physical substrate, the microbiont receive the organic carbon compounds produced by the phycobiont through the photosynthesis, as well as nitrogen and other nutrient derived from the atmospheric deposition and the mineral weathering. Okay. Now, the next question is here. Give a comparative account of the class kingdom fungi under the following mood of nutrition and mood of reproduction. So, according to the mood of nutrition, how the fungal kingdom is being classified, how what are the classes, and according to the mood of nutrition. So, let us see the answer. So, if we talk about the mode of nutrition, then we are having the first one is the zygomycetes. So, zygomycetes is primarily the saprophytic fungi obtaining nutrients by the decomposing matter. So, there are some of the decomposing matter where these zygomycetes are there. So, they decompose the matter and they take the nutrients from them. Some species can also act as a parasite. So some of the species of the Zygomycetes class are parasitic and invading and feeding the living organism or other fungi. They can take food from the other living organism. That could be a plant, that could be animals, and even sometimes that could be the another fungi. So the first one is the Zygomycetes. Next is the Ascomycetes. Ascomycetes. So ascomycetes exhibit the diverse mode of nutrition, including saprophytic, parasitic, and symbiotic. So three mode of nutrition you can see. Whereas in the zygomycetes, we are having two mode, that is the saprophytic and parasitic. So many ascomycetes decompose the dead organic matter, while the other form the mutualistic relationship with the plants, that is the mycorrhiza, or with the sometimes with the algae, that is calling making the lichen association. Hmm? So they are making the association sometime with the higher plants and roots of the higher plants. So that is called as the mycorrhizal association. And sometimes they are making the association with the algal, making the lichens. So this is the symbiotic type of association we can see with the ascomycetes. Now some are notable plants that is pathogen, causing disease such as powdery mildew and Dutch elm disease in the case of plants. So they are showing the pathogenic mode of nutrition as well. Next is the basidiomycetes. If we see the mode of nutrition in basidiomycetes, then we include a wide range of nutritional strategies, including saprophytic, parasitic, and mycorrhizal association. So many basidiomycetes are decomposers, breaking down the complex organic compound in the dead plant material. Okay, so in the dead plant material, we see that they are decomposer. They are acting as a decomposers and breaking the complex organic substances into the simpler form. They are making the complex organic substances into the simpler form. So some are ectomycorrhizal, means they are just making the covering over the roots of the higher plants on the surface only. So they are ectomycorrhizal, forming symbiotic association with the roots of the trees, while others are pathogens of plants and animals. So this is about the basidiomycetes. Now the other type of is the deuteromycetes, that is the fourth class, that is the fungal imperfecti. Now if we see the nutrition in this class, then deuteromycetes encompass fungi with unknown or atypical sexual reproductive structures and are classified based on their asexual reproductive structures. Means the sexual stage is not being reported in some of the fungi, so they all are capped in a separate class, that is called as deuteromycetes. Now they exhibit a various mode of nutrition, include the saprophytic, parasitic, and symbiotic relationship, right? So they are showing the saprophytic mode of nutrition, they are showing the parasitic mode of nutrition, they are even showing the symbiotic relationship. Okay, so a common example include the penicillium aspergillus. Okay, these are some of the common example, Penicillium aspergillus, which are often associated with the decomposition and food spoilage, as well as some human pathogen like Candida species. Now, zygomycetes, mode of reproduction, if we talk about, so we have seen the four different classes which showing the mode of nutrition. Now let us see what type of reproduction they are saying. Showing. So the first one is the zygomycetes. So zygomycetes reproduce both sexually as well as asexually. So both the method we can see in the case of zygomycetes. Asexual reproduction occurs through the formation of sporangia, which releases the spores, And the sexual reproduction involves the formation of specialized structures called zygosporangia, where the fusion of haploid hyphae result in the formation of diploid zygospores. Okay, so this is the diploid zygospores, 2n in number. Next is the ascomycetes. Now, what we see, the mode of reproduction in the ascomycetes. Ascomycetes exhibit the diverse array of reproductive strategies, including both the sexual and asexual reproduction. So, asexual reproduction typically involves the formation of conidia and specialized hyphae called conidiophores, right? So, they are following the asexual reproduction by the formation of conidia, right? And the specialized hyphae, that is on the specialized hyphae, that is called as conidiophores. So a stalk like structure that are having the conidiophores, and on that you can see conidia. These are the conidia. These are the exospores. They are not covered by any type of covering. So they are called as exospores. Sexual reproduction involves the formation of a specialized structure called asci. So if we see the sexual reproduction, then you can see a sac-like structure in which the spores are present in this way, and these are called as ascospores. Ascospores. And this particular sac, which is the covering this ascospores, is called as sky or ascus. Okay, so which contain the haploid ascospores produced through the process of meiosis. Next is a mode of reproduction is the basidiomycetes. So basidiomycetes reproduce both sexually as well as asexual. Okay, so both the method are, we can see in the case of basidiomycetes, sexual method as well as asexual method. So asexual reproduction occur through the formation of specialized cells called conidia or through the fragmentation of the hyphae. So either they are doing the process of asexual reproduction by the process of formation of asexual spores, that is called as conidia, or even by the fragmentation of the hyphae. Now the fragmented hyphae can give rise to a new whole individual. Sexual reproduction involves the formation of basidia. Now this is the basidiomycetes. Okay, so they form the sexual spores, that is called as basidia, which produced by the haploid basidiospores through the process of meiosis. Okay, the next one is the deuteromycetes. Fungi imperfecta. This is very, very important. Deuteromycetes are characterized by the absence of known sexual reproductive stage. Okay, they are not showing any sexual reproductive stage, and that is why they are called as fungi imperfecti, imperfect fungi. So they reproduce solely through the asexual means. So only asexual stages has been reported in the case of deuteromycetes, and primarily through the production and the dissemination of conidia. Hmm? So we can see they are producing the conidia as an asexual spores. They spread these conidia in the environment, and by that, they can give rise to a new individual. So it means that the asexual reproduction is taken by the means of conidia. So sexual reproduction is either absent or not well understood in this particular group. So in summary, while there is some overlap in the nutritional and reproductive strategies among the classes of kingdom fungi, each class exhibit a unique characteristic in terms of how they obtain nutrients and they reproduce. So these two things are very, very important in the case of fungi. Okay, so understanding these differences is crucial for studying the ecology, diversity, and importance of fungi in the various ecosystems. So let us see the next question. Which are the characteristic features of euglenoids? Okay, not which, what are the characteristic, means you have to tell the characteristic feature of the euglenoids. So here comes the characteristic features. The typical features of euglenoids are absence of cell wall. So they are protista. Euglenoid are placed in protista. Okay, protista. This is a one of the kingdom according to the Whitaker classification. Okay, So Protista is one of the kingdom in which this euglenoids we see or we keep them. And what is the characteristic? The first one is they are not having any cell wall. Their body is flexible because there is a protein-rich layer called pellicle. If you see the euglenoids, okay, then their body is covered with a pellicle covering. This is a very, very important characteristic of the protistans. Pellicle. This is the pellicle. And the two flagella different length are found. So here you can see one is the larger flagella, and this is the smaller flagella. Two flagella are there. One is larger and one is smaller. They are autotrophic in the presence of sunlight and heterotrophic in the absence of sunlight. So they are having chloroplast in them, but they are not having cell wall. This is a very unique feature. Okay. So euglenoids are not having any cell wall, but yes, they are having chloroplast in them and they can perform the process of photosynthesis in the presence of sunlight, but in the absence of sunlight, they cannot make their own food. So they are also showing the heterotrophic mode of nutrition. Next question, give brief account of viruses with respect to their structure and nature of the genetic material. Also name 4 common viral diseases. So, structures of virus, genetic material. The viruses can have their DNA, deoxyribonucleic acid, or RNA, ribonucleic acid as their genetic material. So, their genetic material could be DNA or RNA. And this genetic material carries the instruction necessary for the viral replication and assembly. So for the process of replication, there is a requirement of the nucleic acid. So this DNA and RNA could be the genetic material for them. Next, you see in the structure of virus, the protein coat that is covering the nucleic acid. So surrounding the genetic material is the protein coat that is called as capsid, which provide the protection and shape to the viruses. And the capsid may be composed of one or more protein subunit that is called as capsomers arranged in the various symmetrical pattern. If we see the envelope, then some viruses have an additional outer layer that is called an envelope, and which is derived from the host cell membrane during the viral replication. The envelope may be contain the viral glycoprotein, which play a role in the viral attachment and entry to the host cell. So this is very, very important for their attaching to the host cell. DNA viruses. DNA viruses have a double-stranded, single-stranded DNA as their genetic material. And for examples, herpes virus. So herpes simplex virus is an example. And if we talk about the adenovirus, then adenovirus is an example. And papilloma virus, then human papilloma virus is the example of that. So these are some of the examples. If we talk about the RNA virus, then RNA virus have their either single-stranded or the double-stranded RNA as their genetic material. And examples include influenza virus, influenza A virus, human immunodeficiency virus, that is HIV, and hepatitis C virus, HCV. Common viral diseases, if we see, then influenza is one of the disease. Influenza is a highly contagious respiratory illness caused by the influenza virus. And symptoms include the fever, cough, sore throat, muscle ache, and the fatigue. And influenza virus belong to the family Orthomyxoviridae. Another disease, that is the common cold, we see in the case of this is again a viral disease. So common cold is a viral infection in the upper respiratory tract. It occurs in the upper respiratory tract, typical caused by the rhinovirus or the coronaviruses. And symptoms include the sneezing, runny or the stuffy nose, sore throat, and cough. The last one is the human immunodeficiency virus, that is, you all know, that is causing the acquired immunodeficiency syndrome. So this HIV is a retrovirus that attack the immune system, specifically targeting the CD4 of the T cells. And HIV infection can lead to the acquired immunodeficiency syndrome AIDS and condition characterized by a weakened immune system and susceptibility to opportunistic infections. And they belong to the family Retroviridi. So this is all about this session. We will have some other questions that we will discuss in the next session. Till then, thank you very much.